Welcome to our current lecture. It's the fourth one that we have for SCP-105, Life in the Universe. So far, we've been exploring the different locations in the universe where we think we might be interested in looking for life. But when we first started off looking at the grand scheme of the universe, became clear that the first thing we would need to do is figure out what we were looking for, what life really is like. And that was a difficult question to try to answer. So we started by looking at atoms and then moved on to some of the most important molecules for life, including amino acids, which build proteins, and nucleic acids, which are involved in genetics, DNA, and RNA. DNA stands for deoxyribonucleic acid, a very important molecule in our existence. And its associated nucleic acid is RNA, ribonucleic acid. DNA has two strands that wind together, forming a double helix, while RNA is single-stranded. That makes RNA a little bit less secure. It breaks up a little bit more easily. It doesn't store the genetic information as easily as DNA or as securely as DNA does, because there's only one strand. It can break apart easily. But we're going to come to understand that this RNA might be a more important chemical for understanding the origin of life than DNA. And in fact, right now, it is the theory that most scientists are interested in for explaining how life may have emerged from non-life. So let's get started in exploring this subject. I will start showing some slides that will take us from looking at some of the more complex chemicals you have been creating in this class to what eventually will show up as the first life forms on Earth and where they might live, whether that might be in environments that we might recognize as being environments that are places where life exists today, or whether they might be very strange environments, and whether these life forms may be very different and not at all what we might expect. And this should be an important thing for us to understand, because as we look for life out in the universe, we may find some environments that are very different from our own. And so understanding the strangest places that life can exist here on Earth might help us to prepare for discovering life in even more and stranger environments and more interesting places out in space. So I'm going to start with a discussion of this strange idea, which people have been thinking about for some decades now, as to the origin of life on Earth. One of the remarkable things about life today is that all life that we know of originates from other living things. But as we go back in cosmic time, we see that what the most common forms of life were in the past aren't necessarily similar to the forms of life we see today. There has been a process of change that has happened over millions and even billions of years, which has resulted in living things that we often recognize as being alive: animals and plants and fungus. But when we actually look at what the most diverse forms of living things there are, the microorganisms in the world, we find that those things look very different than us, much simpler than us. And in fact, some of these microorganisms are not much different than small membranes filled with chemicals. As we go back in time, the evidence is that that was the predominant form of life on Earth for hundreds of millions, if not billions of years, very early on. When we go back 3 and 4 billion years into the past, that was what life looked like. Imagining a human being coming from individual atoms and chemicals is difficult. But figuring that a small microorganism, which is a membrane filled with chemicals, might emerge from non-living chemicals, nucleic acids, amino acids, and proteins, this is perhaps an idea that's more interesting and easier to come to grips with. And so people who study the origin of life from non-living things, in particular the chemicals that aren't alive but are important for life, to a membrane or a cell filled with chemicals that we would recognize as a living thing, people who study that process have come up with some interesting observations that, when we put them together, might tell us how this actually occurred here on Earth. We aren't certain that this is the process. This is very much a hypothesis, the hypothesis called the RNA world. But it is an intriguing one, and it is one that is studied a lot. And it may be that a different hypothesis is the correct one. We may learn that in the future. But some of these processes are so intriguing that it seems likely that they will show up in whatever we find to be the ultimate explanation for how life emerged. Incidentally, this question of the origin of life has a name, and the name is abiogenesis. A means not, and bio means life, so not living. Genesis, the start of things that are living from things that are not living, abiogenesis. And the hypothesis we're going to look at most carefully is this one you can see here, called the RNA world. The RNA world is a hypothesized time which would have occurred about 4 billion years ago on planet Earth, right after the planet had cooled after its initial formation. When planet Earth formed, it was very hot. As things coalesce in any sort of formation of stars or planets, and they collapse down to the size of the planet or the star, they start out very, very hot. But that heat quickly dissipates and things begin to cool down. Once the surface of the Earth was cool enough to allow for liquid water, and many of the processes we know are important for living things today, there wasn't immediately, as far as we know, life everywhere. But there were a lot of the atoms and molecules and precursor chemistry necessary for life to form. And there were experiments, and continue to be experiments, where people look at what the environment of that early Earth looked like, with the chemicals that may have been there and the processes, and see whether more complicated molecules, such as the amino acids and the nucleic acids that you built models of in the laboratory, could occur and exist in those environments. One of the questions that people have who look into this is whether RNA, ribonucleic acid, could have formed. Because if RNA can form in the early part of the world, in the earliest stages of our Earth's development, well, then many of the processes of life come along with it. And the reason we know that can happen is if we take RNA by itself in a laboratory and look at what can happen, just as RNA interacts with only itself and water and some other non-living chemicals, a number of intriguing things can occur. One thing that can occur is that RNA can actually replicate itself. Now, this sounds very surprising to many people. We think of replication and reproduction as something that is fundamental to living things. But there are chemicals out there which can reproduce themselves, and RNA happens to be one of them. The action that takes place with RNA replicating itself still occurs in the cells in our own body. It occurs in a part of all of our cells called the ribosome. And this ribosome is really just a location, a chemical, that can allow for RNA to replicate itself by having RNA exposed to the right other sorts of chemicals. It has been observed that RNA can do this ribosome action in environments where there are no living things at all. This is very intriguing because if RNA can self-replicate, it's already doing some of the processes that we said life needs to do. The other thing that RNA can do is attach itself to amino acids. In fact, this attachment to amino acids is exactly how we build proteins in our own bodies. DNA has the recipe book for how to build proteins. And the DNA travels through the ribosome complex, and the RNA attaches to the DNA on one side and attaches to a protein on the other side. We call that tRNA transcripting RNA that allows for the production of these proteins. Well, in our cells, this happens near the ribosome. But if you have amino acids that are in the environment, and we know that the early Earth had the right chemicals to form amino acids without living things, they do form that way. Those amino acids can attach to the RNA and form simple proteins. So the idea in the RNA world is that there weren't living things, but perhaps there was quite a lot, or at least a high enough amount of RNA to allow for both of these processes to occur. For RNA to replicate itself and for RNA to allow for the production of simple, but still very important proteins. So as RNA allows for proteins, eventually another nucleic acid enters onto the scene, DNA. DNA is a bit more fragile than RNA. It needs to be protected. But it does have the advantage of not being as sensitive to outside changes. It stores information a little bit better than RNA does. So once you introduce DNA into the system, you have the beginnings of the processes that happen in our cells, in all bacterial cells, and really every living cell on the planet involves DNA encoding RNA and RNA encoding DNA, and RNA also pulling along proteins to synthesize. And we think of that as being the fundamental process here in living things. So during this time of the RNA world, there weren't proteins that were engaged in the biochemical reactions yet, but RNA was able to begin that process. During the RNA world, RNA had many different roles. It could store information, and it could also transfer information And it could even allow for different forms of chemical reactions. In our bodies today, many of the chemical reactions inside of cells are mediated by proteins. They're catalyzed or given enzymatic action by proteins. But those sorts of catalyzed reactions can occur with just RNA as well. So RNA has this amazing character where it can do a lot of the different things that living things require, and in our bodies are done by chemicals other than RNA. But RNA can also do it all by itself. This is why the RNA world hypothesis has gotten a lot of interest, because the processes that are necessary for living things seem to be done by RNA by itself. Eventually, what happened in this model is that all of these processes began to occur in the same sort of area. And a membrane formed around the RNA, the DNA, and the proteins, forming the first what we would call protocell. The border between life and non-life is established for many biologists right there. And once you have a cell, that forms the end of the RNA world. We no longer live in an RNA world. We live in a world of life. This is another way of looking at what that might seem like. You start with an Earth that does not have any living things in it, but does have RNA. Proteins and RNA are made, as it says in this slide, abiotically, without life. RNA is able to self-replicate, and it's able to have chemical reactions that occur that bind it to proteins and also allow it to transfer information from one place to another. However, there is some process where that RNA gets encased, perhaps with DNA and protein as well, inside a cell, as it's listed here, a lipid or lipoprotein vesicle. Lipids are a form of chemical that are in the cellular membranes of every living thing. And they form in many different ways. And if you can combine that with a nucleic acid and amino acids, this is what we call a cell. So that border between life and non-life becomes a little bit more fuzzy. Now, we haven't seen that process happen in the laboratory. We haven't seen a cell form from non-living things. This is a hypothesis that this can happen. However, all of the intermediate steps have been seen. And it may be at some point that this does occur in the lab, given the right conditions. People are still investigating. The idea is that once you have early cellular life, that then proteins and RNA interact more and more inside of cells, and eventually DNA becomes the storage of the information that the cell needs to make the proteins and allow for the reactions that allow for life to exist and thrive in different environments. This is what is called modern cellular life at the bottom of this slide. At some point, RNA no longer has all of the roles that it once did. The replication of the genetic material is done by DNA. The different reactions, chemical reactions that occur inside the cell are mediated by proteins instead of RNA. But RNA is still there, acting as a messenger and moving things around in the cell. So this is the idea that many people have come up with and have seen different steps that can occur in the laboratory that might explain how life emerged in an early Earth. But what was the first life in the early Earth, and when did it emerge? Well, up at the upper left, we have some of the earliest pieces of evidence for life on Earth. This isn't life itself. This is a crystal, and inside of this crystal is preserved carbon. Now, living things use carbon a lot. And one of the things that living things do with carbon is they separate out the different kinds of carbon. In particular, there are two different kinds of carbon that living things might be using. And one is called carbon-12. That's carbon with 6 neutrons and 6 protons. And one is called carbon-13. That's carbon with 6 protons and 7 neutrons. Living things prefer carbon-12 over carbon-13. And we can see that when we look at the carbon inside of living things and compare it to the carbon that comes in meteorites from space. Living things preferentially concentrate that carbon-12. Well, when we look inside of this crystal at the top left, what we find is a concentration of carbon. This was carbon that was in the environment some 4 billion years ago. And we find a different ratio of carbon-12 and carbon-13 than we do in the other material that comes from earlier times or comes from places where we know life doesn't exist, such as rocks hurtling through space. Scientists wonder whether this is an indication that life was already around some 4 billion years ago. In fact, many think that this is strong evidence that that's the case. This is just looking at atoms. This is not looking at actual living things. But since we know living things can sort atoms in this way, it's intriguing evidence that living things might have been around as early as only a few 100 million years after the formation of the Earth. Well, on the lower left, I'm showing you the first fossils that are known of any living things. These are called stromatolite fossils. And although this may not look like living things, we can recognize these forms as being very similar to living things we see here on Earth today, formed by a type of microorganism called blue-green algae. There are different kinds of algae, which are these small single-celled organisms that dominate water. And different forms of algae that are known include different colors. But the oldest form of algae, the one that has been around the longest, is this blue-green algae. And it forms these very distinctive layers that you see in this fossil on the lower left. These layers are seen in the oceans of today, and they're also seen in these ancient fossils. So we know that life has been around for at least some 3.5 billion years. But according to the composition of the crystals up in the upper left, the zircon crystals, probably before then as well. Now on the upper right, we're looking at some of the first fossils we have of organisms that are fully intact. We call these things microfossils. And you can see the measurement here is perhaps a tenth or maybe a few tenths of a millimeter. Very, very small measurements here. And these microfossils are showing us some forms of bacteria and other microorganisms that existed in the early Earth. These are very difficult to discover, but they have been looked at very carefully because we expect that if we do discover fossils on other worlds, they might take this form. Well, the stromatolites and the different microorganisms that existed billions of years ago did a lot of interacting and did a lot of things and had to cope with a lot of changes in the environment. There were periods of time in the early Earth's history where things became very cold, and we have evidence that ice actually formed all the way to the equator of the Earth, covering it, as you see in the lower right. This is called the snowball Earth, and it occurred at least 2 times in the history of our planet. Imagining that ice covers the entire planet might make you think that it would kill all life, but it didn't. This is probably due to the fact that ice floats on liquid water. And so a lot of these microorganisms that live in liquid water could live beneath the ice. And we'll examine some microorganisms that continue to do that to this day. Another very important event that occurred early on in the history of life on Earth was something called the Great Oxygenation Event. It occurred about 1 billion years after life appeared. And when this event happened, the entire atmosphere of the planet changed. Early Earth's atmosphere had a lot of carbon dioxide and nitrogen in it. But one of the things that life did was take that carbon dioxide, break it apart, and release oxygen, changing the CO2 into O2. Now, we should be very grateful that this process occurred because we breathe that oxygen today. But early life required that carbon dioxide to survive, and as it disappeared and was replaced by oxygen, much of that life was subject to oxygen that actually would cause it to be poisoned. There are still microorganisms on our planet today which cannot live in an oxygen environment. We find them in parts of our planet where oxygen is scarce, at the bottom of the ocean, for example. We think that a lot of those microorganisms are the living fossils of these microorganisms that once dominated the planet and killed themselves by changing the atmosphere. So living things had to adapt to living things changing the planet. And of course, one of the things we like to think about, or perhaps are concerned about, is that we again are changing characteristics of our planet, perhaps making it difficult for ourselves to survive. Well, after this oxygenation event, things changed rapidly. And one of the things that changed was the kind of cell that existed in planet Earth. For most of the history of our planet, the kinds of cells that existed on Earth were called prokaryotic cells, in what we call bacteria. You can see examples of them on the left. There was nuclear material on the inside, we call it nucleic acid, and amino acids on the inside of a cell. That's covered by a membrane, and perhaps there were some other things extending out, things called pili and flagella. But for the most part, bacteria were fairly simple, bags of chemicals. Now, some of these bacteria became very specialized, and as they became very specialized, they began to do various processes like take carbon dioxide and turn it into oxygen. Eventually, 2 or more of these bacteria would come together and start to work with each other. And at some point, they became part of a larger and more complicated cell. Cells that have different parts, which have their origins in past bacteria that have come together to work together, are called eukaryotic cells. And you see an example of them on the right. Almost all of the things that we recognize today as being alive, all of the multicellular organisms that are on our planet, anything that has more than one cell, has cells that are of this form, eukaryotic. They have different parts to the cell. There are parts of the cell called the nucleus, where the nuclear material is kept. There's a part of the cell called the mitochondria, where a lot of the energy is produced for the cell to use. Mitochondria have its own nuclear material, its own nucleic acids. And this indicates that this was once a separate organism that then got incorporated into eukaryotic cells. In plant cells, which are also eukaryotic, there's another type of, we call these things organelles, called chloroplasts, which are what convert carbon dioxide into oxygen and provide food for the plant. And this is probably also an ancient life form that got incorporated into the cell. Chloroplasts also have their own nucleic acids. And you can see a lot of these other parts to eukaryotic cells. If you take a biology class, you learn all about these parts of eukaryotic cells, as they're very important for all of the processes that we associate with the more complicated life, what we call complex life. And this complex life really starts to emerge only about 500 million years ago. So for billions of years, single-celled organisms were all that was. And then suddenly we started to have multicellular organisms made up of these eukaryotes. This is showing the amazing abundance of living things here on Earth and how we begin to categorize them. We start off with three big domains of life that are highlighted in blue at the top. We've got bacteria, we've got eukarya, and we've got archaea. Bacteria are by far the most common life forms on our planet. They have the most genetic diversity. If you were to look at a random nucleic acid that you find on Earth, chances are it's from a bacteria. But other forms of life emerged and are related to these bacteria as you go back to earlier and earlier times. And some of these forms of life have many cells and have very complicated cells and parts of their cells, and they call that the eukarya. And there are three different eukarya that we find are extremely important as they form the largest forms of life on the planet. And those are the plants, the animals, and the funguses. And you can see those at the top. Those are called the crown eukaryotes. These are the things that are most closely related to each other. This diagram is made, by the way, by looking at how close the DNA matches between different organisms. 70% of the diversity of DNA in living things is found in bacteria. And another 20% is found in another part of the domains of life we call the archaea. Archaea are very strange forms of life, hard to find, usually in places where you wouldn't expect to find living things. They're more closely related to eukarya than they are to bacteria. So we are closer related to them in terms of our genetic distance, which is a term we use to describe how different our genetics are from one organism to another. They're closer to us than they are to bacteria, but they're also very bizarre, and they seem to harken back to an older time. Perhaps they are the precursors to the living things that we are today. Now, on this diagram as well, you'll see a number of different color codes where there are different kinds of organisms that can live in different environments. One of the environments that they can live in are environments called environments for thermophiles or hyperthermophiles. These are environments that are much hotter than normal life can live. Thermophiles and hyperthermophiles can live sometimes in water that's nearly 100 degrees Celsius, close to boiling. And you might find these in ocean vents at the bottom of the sea. We also find psychrophiles, those things that are colored in blue. And psychrophiles are things that can live near freezing and sometimes even below freezing. We find piezophils being things that can live under high pressure. Halophiles, these are organisms that can live in salty environments. pH extremes include the acidophiles and the alkalinophiles. These are the living things that can live in acid environments, the living things that can live in basic environments. These are chemicals that are often dangerous to living things, but some living things require an environment that's much more acidic or much more basic. And then finally in yellow, you see the radiation resistance. These are the microorganisms that can resist high amounts of radiation, things that would normally kill other things. So these are what we would call extremophiles. And extremophiles are really important things for us to understand and study, because extremophiles are exactly the sorts of living things that might be discovered outside of the Earth, where the environments are different from the environments we see here on Earth. So you can see that many of the extremophiles are bacteria and archaea, single-celled organisms that would look very different from us. And you can see where they might be in extremely bizarre environments that would have high levels of acid, high levels of heat, sometimes much radiation. Here's an example of an environment where you might find extremophiles. This is the bottom of the ocean at an ocean vent. At the bottom of the ocean, volcanoes that occur can superheat the water. And as they superheat the water and release what we would consider toxic gases, you might expect there to be no living things. But as you can see in this image, there's actually a tremendous amount of color down at the bottom of the ocean. This is indicating that there are some living things. These are animals that are living at the bottom of the ocean. They have no exposure to sunlight, which means they're getting all of their energy from the volcanic interactions themselves. And the question we might have is, what sorts of living things are able to feed off of that energy? The answer is that there are these extremophiles that can live in extremely high heat and will absorb the nutrients that are coming from these ocean vents and actually thrive and cause these entire ecological groups to form, all the way up to complicated animals. Here's another view of those ocean vents with some of the superheated water zipping up out of them. And surrounding them, very close to where it's hottest, are amazing numbers of living things. Here's a close-up of some of those red objects you saw in the previous image. These are some of the tube worms that you see at the bottom of the ocean that survive off of eating some of these extremophiles that we will look at. And the extremophiles are often single-celled organisms. Here are some thermophiles, which live right at the edge of these hot ocean vents at basically boiling temperatures. Not only do they survive, but they actually require that level of heat in order to survive. If you took these thermophiles and put them in a room temperature water, they would die. Here's another place where people explore and look for extremophiles. This is actually Yellowstone National Park, the western part of the United States. This is a lake that you can walk past, and you can see there is a path that runs in the lower right. to give you a sense of scale, if you look carefully, you can see some people on this path. So it's a fairly large lake, maybe the size of the reservoir in Central Park. But what's different here is that the lake's water is heated enough so that you see steam coming off of the lake. This is because Yellowstone is actually a large volcano. But more than that, there's also toxic, what we would consider toxic anyway, gases coming from that volcano. You might expect then that there would be nothing alive around there, but you would be wrong. In fact, you can see some yellows and greens surrounding the superheated and extremely odorous, actually stinks, sulfur pond. And those yellows and greens are actually microorganisms, extremeophiles, that can live in highly acidic and high temperature environments. And there are also some that can live directly on salt. This is salt, the edge of one of those ponds, and the yellow that you see is some form of microorganism that can live in extremely salty environments. Halophiles is what we call them. So even in the places where we might expect to find no life here on Earth, we find a tremendous amount of life. And that might indicate to us that when we go looking outside of the Earth, we might expect that there might be no life, but in fact, the living things might be there in an environment where we would not even expect them to be. Here's another example of such a strange and environment where we would die almost immediately, but we find lots of living things. This is actually a lake in Antarctica. This lake was explored even though it was beneath some kilometers of ice. And what was done to explore this lake, which hasn't been exposed to anything outside of the water in that lake, covered in those kilometers of ice for millions of years, is to drill all the way down. This is a drill that's at the top on the ice that's above the lake, and it drills down into the bottom. Here's how you can see it, drilling down 4 kilometers. The name of this lake is Lake Vostok, and it was explored after this drill was sent down into the lake. And surprisingly, there's a significant amount of life that's there. This life hasn't been exposed to sunlight for millions of years. And so very strange things have happened. There are still some fish that survive in this lake. They've become transparent and their eyes don't work because there's no sunlight. And there are also some microorganisms that can live near the ice in this lake. And these are extremophiles. These are the psychrophiles that can live in extremely cold environments, basically in frozen ice. And they eke out an existence by going through the little channels in the ice. And they can live in extremely cold environments. And if it warms up even a little bit, these microorganisms don't survive. Well, we're going to explore another microorganism that's not an extremophile, but it is a microorganism that can survive in a lot of extreme environments. This microorganism is called a tardigrade. Sometimes people call them water bears. And you can see in this comic here that they have fascinated a lot of people. Now, these are animals like you or I. They're multicellular. And they can grow to be fairly large because we have a strange view of what small and large is at this point. When we talk about small things, sometimes we're talking about things that are easily a millionth of a meter, right? A micron or a micrometer in size. Well, tardigrades can be up to half of a millimeter. So some 500 times bigger than that smallest measurement we were talking about here. That's still very small, right? It's just barely the size we could see with a human eye, but with a relatively small microscope, and if we know where to look, we might be able to discover some of these. They survive in all sorts of environments, but they especially like mosses and lichens. And what happens when it dries out is they curl into a little ball and go into something called a tun state, where they look more or less like a grain of sand. It's only when they're exposed to water do they animate, come alive, and start to move around. And you can see these pictures of them with things that have shapes kind of like feet, but are not really feet, and things that look kind of like a mouse, but aren't really a mouse. They don't have eyes. They don't have any of the anatomical features you or I recognize, but they're fascinating animals. They've been shown to survive in all sorts of environments. They've been shown to survive if they've been frozen for a bit and then thawed. They can survive after they've been boiled for a bit. They could be suspended in this tun state, this sort of state of almost like a grain of sand, for some months, at least, in some cases even years. You can expose them to high amounts of radiation and they survive. They've been sent to space, and when they come back, they've been shown to survive. There's a lot of different environments that these small little organisms can survive in that human beings, for example, cannot. They might make for good astronauts. This is an image of a tardigrade. Now, this image is enhanced in the sense that the color is added to kind of highlight some of the features. Tardigrades are not opaque. You're actually transparent. You can see right through them. But this image was taken with a special microscope that can see the very tiny details on them and enhance them. That's called a scanning electron microscope. And so when they color coded these things, they showed the pseudopods, which are the sort of feet-like protrusions, and then the mouth. And they made the body opaque. And you can kind of see this as a tardigrade. The tardigrade is in its environment here. It's near some moss where it was found. Here's another picture of a tardigrade in the same color coding scheme near some moss. Picture taken with a scanning electron microscope. And yet one more that was taken with a scanning electron microscope. Now you are going to be looking for these. You've already collected the samples that you need, and hopefully we will find some. Sometimes it's hard to find tardigrades, but we've usually been successful. And so that's what we will look for. We will look for some of these that may have survived the freezing of the winter months. We'll find some that have been exposed to the dry environments on the outside, but then once they're exposed to water, are reanimated and can start to wander around. And think about whether these tardigrades might be able to survive some trips that would otherwise kill other living things. So this is our exploration of the strange and wild variety of living things here on Earth. We'll continue our explorations, but we will reach quickly a limit to our knowledge. We've never discovered living things outside of the Earth. Will they look like extremophiles? Will they look like tardigrades? Or will they look like human beings? Or will they look completely different from all of us? Maybe they won't even use the chemistry that we've been studying. Maybe they won't have cells. Maybe they won't have organelles. It's hard to know what the unknown is, but we'll continue to look and hopefully we'll start to find. So thank you very much. I hope you learned a lot about these different forms of life here on Earth. And I look forward to talking with you next week about more questions about where we might look for life out in the universe.